So in the previous video we were discussing about the location of the enzymes, the site of the enzymes where they work. So these are the intracellular enzymes and then the extracellular enzymes. The extracellular enzymes are those enzymes. Pepsin is an enzyme which is working in the stomach cavity. So actually they are released in the stomach, and that is why they are outside the cell in the cavity of the stomach, and they are working. Now all enzymes are catalyst. All enzymes are no. See, the first statement is that all enzymes are catalyst, and the second statement is that all catalysts are enzymes. So statement second is incorrect. Why? Because all the catalysts in the body are not enzymes. But yes, all enzymes are catalyst. Now we come towards the characteristics of enzymes. See, you must know about the history of the enzymes, that Wilhelm Kuhne was the first German physiologist in 1878 who used the term enzyme. These are what? These are globular proteins. Globular means they are in a circular structure and they are proteins. Like all proteins, they are made up of long linear chain of amino acids. So they are made up of linear chains of amino acids. They have a 3D molecule, like a three-dimensional molecule, and all biochemical catalysts are not proteins. For example, are some RNA molecules also catalyze reaction, but these RNA molecules are not proteins. So this statement was that all the catalysts are enzymes. Yes, but all no the they say that all catalysts are enzyme. That was incorrect. This is the example of this, that RNA molecules is what? It is a catalyst. Yes, it is a RNA is catalyst, okay? But this catalyst is not an enzyme. Now you get my point. But yes, all enzymes are these all all the enzymes are catalyst, yes. But all the catalysts are not enzymes. RNA is a catalyst, but RNA is not an enzyme. Understand? RNA is not an enzyme, but it is a catalyst. All the enzymes are catalyst, yes, but all the catalysts are not enzymes. So now we come towards the characteristics which are very important. As your syllabus is reduced, you must learn all the topics, because now all the topics become important. Now the first point is almost all enzymes are proteins. Yes, they are made up of amino acids. So actually what happens is that if these are smaller blocks of amino acids, they make up to form what? Larger chains of amino acids. How? Larger structures of amino acids. Understand? Now amino acids are like this. They are very long chains. Understand? These smaller particles join together to form larger chains. Understand? Now second point is that most enzymes are proteins and they are made up of amino acids. Second point is that most enzyme reactions, there are millions they are millions of times faster than those which are uncatalyzed. What does the enzymes do? What does the enzymes do? What do the enzymes do? They catalyze reactions. Okay, with all the catalyst, enzymes are not consumed with the reaction they catalyze. This you will understand when you will go through an example. See, when there is a reaction going on, what happens is that First of all, you have a substrate, understand? Then you also have enzyme here, understand, before the reaction. Now what happens is that here you have a product, and again you have the enzyme which is unaffected. Understand, enzyme is not used, it is unaffected, it is increasing the reaction, and it is not consumed by the reaction. Understand, it is increasing the rate of the reaction. What do you mean by the term catalyze? What do you mean by the term catalyze? It means increasing the rate of reaction. Understand? Now, the third point is that enzymes are usually very specific for the type of the reaction and for the nature of the substrate. For example, if the enzyme is specific to a fat molecule, they will only go towards the fat molecule, they will bind to with it, and then the reaction will continue. Understand? Next point is that, so actually they are specific for the reaction and for the nature of the substrate also. They will carry out a special reaction with a special substrate. For example, this is reaction number A, so it has a specific enzyme. Now there is reaction number B, different enzyme. Understand? Now reaction number C, this reaction has a different enzyme. So actually different reactions, different enzymes. Only a small portion of enzyme molecule is directly involved in catalysis. Catalysis. This catalytic reaction region is known as active site. It recognizes and binds the substrate and then carries out. Actually, these are just the terms you learn. You will actually come to understand these terms when we'll be studying further. But you just go through and listen to the idea. This topic gives you the idea. We'll be discussing them in detail in the next topic. So they say that the enzymes, they have an active site which is involved in the catalysis, and which means it is involved in the reaction. The other part is not involved. It recognizes and binds with the substrate and then it carries out the reaction. Enzyme production can be enhanced and can be diminished. Diminished means decreased. Increased or decreased in the production of enzyme is according to the need. If you need the enzyme in a larger quantity, the body will secrete the enzyme in a larger quantity. If you need it in the lesser quantity, if the substrate is more, the enzyme will be more. If the substrate is less, the enzyme will be less. Understand? Now they are explaining the inhibitors and activators. What are inhibitors? They actually decrease the enzyme activity, and activators actually increase the enzyme activity. So the first option is the quantity will be controlled by the cell, more or less. Again, second option is we can have inhibitors which can come and block the reaction, and activators which can activate the reaction. Okay, that again you will understand later. Some enzymes do not need any additional component to work, but some, yes, they do need. These components are known. See, all these terms are very important. What are inhibitors? What are activators? What is activation energy? What is active site? What is anabolism? What is catabolism? And now we come towards the term which is the cofactor. So some molecules and ions which are non-protein in nature, they they do what? They can be organic and they can be inorganic also. But inorganic and organic, uh, in both the cases they will be non-protein. Understand? They will not be a part of protein structure, they are not in protein, but they can be organic and they can be inorganic. If they are so actually cofactors, see the cofactors, these are the cofactors, and the cofactors again have two types, inorganic and organic. Okay, so the example of the inorganic are the metal ions. They do what? some of the enzymes require cofactor. The cofactor can be inorganic, for example metal ions, and they can be organic, for example flavin and heme. These are the parts of the um, these are present in the body, so they can be organic also. The organic the cofactors, if they are tightly bound to the enzyme, they are known as prosthetic groups, and if they are loosely attached with the enzyme, they are known as coenzymes. So on the basis of attachment, they are prosthetic groups and coenzyme, and on the basis of inorganic and organic nature, again they are differentiated. Coenzymes, they do what? They transport the chemical groups from one enzyme to another. Some important vitamins are riboflavin, thiamine, and folic acid. They act as coenzyme. They do what? They transport chemical groups from one enzyme to another. So they attach themselves to the enzymes and they transport some groups. They provide the enzymes with some groups. Now, several enzymes can work together in a specific order, creating a metabolic pathway. In a metabolic pathway, one enzyme takes the product of the another enzyme as a substrate after the reaction, and the product is passed on to the next enzyme. So that again we can study. This is a very easy concept. Okay, for example, here you have product substrate A. Understand? With after the reaction, what happens is that it changes itself into substrate B. Now this B will be, yes, this here it is the product. Here B is the product, understand? But as we are talking about a metabolic pathway, what happens is that this B can act as a substrate and a certain enzyme can act on it, and then it can it can produce and it can form product C. So actually here for this reaction B is a product, understand? And for this reaction for this reaction B is a substrate, and for this reaction B is a product, understand? So where are the enzymes? The enzymes are two different enzymes. Here for example we have enzyme 1, here for example we have enzyme 2. So for enzyme 2 it acts as a product, and for enzyme for enzyme 1 it acts as a product, and for enzyme 2 this same chemical it acts as a substrate. This same molecule, we'll term it as molecule, not chemical. That same chemical acts as a substrate. Okay,